So we can't mutate human genes, and for the same reason, we also can, at least for the purposes of research, add transgenes, or genes from other species, to humans. And this makes it harder to study human genes. However, we can add them to model organisms. Why would we do this? Well, for example, sometimes we want to know what tissues or organs is a given gene expressed in. In one approach, the GFP gene, which is a gene from a species of jellyfish, is used. This gene encodes green fluorescent protein. You can see the part that gives this protein its green fluorescence in the middle. The GFP gene is often attached to another gene of interest. And when the resulting gene is expressed, the GFP protein ends up attached to the protein encoded by our gene of interest. And we call this a fusion protein. And wherever our gene of interest is expressed within the organism, whether it be the tail, the eyes, or the hair, the GFP protein will also be made. So cells and tissues expressing this gene will glow green under ultraviolet light. In this frog, the GFP gene is attached to another gene, and the only green glow that we see is in the eyes. So from this we can infer that this gene is expressed exclusively in the eyes, and we might further hypothesize that this gene has a function in sight. So we can see here how transgenes, for example GFP, help us to determine where in an organism genes are expressed. And this is just one example of how transgenes are used in research. Now, both humans and model organisms have some traits where only a few distinct phenotypes are seen. Why is this helpful? Well, recall that Mendel studied flower color, and flowers of each plant were either purple or white, but not really any shade in between. These two easily distinguishable phenotypes allowed him to come up with his two laws of genetics because he was able to count the purple and white offspring. These easily distinguishable phenotypes in model organisms allow us to, as Mendel did, study the inheritance of genes. This would have been difficult if the difference between purple and white was not so obvious. This also helped Morgan, of fruitfly fame, carry out gene mapping for the same reason. Until 2002, a major drawback of research in humans was that we didn't have the entire sequence of the human genome, which meant we didn't even know about most of the genes that humans had. As you saw previously, if we know the DNA sequence of a gene, we can use the genetic code to determine the amino acid sequence of the protein that that gene encodes. But if you haven't sequenced the genome, then you don't know the DNA sequences of genes, and you won't know the protein sequence either. This means that you can't compare that protein sequence to that of other proteins to try and figure out its function. Now that the human genome has been sequenced, this isn't a problem anymore, and all the model organisms have been sequenced as well. So once again, if we know the DNA sequence of a gene, then we'll know what the sequence of the RNA will be once it's transcribed, and because we know the genetic code, we can infer from the RNA sequence what the amino acid sequence of the protein will be. And we can then use the amino acid sequence to compare that protein to other proteins with known function and start to get an idea of what that protein might be doing in the cell. And now that we've sequenced the human genome, the genome of model organisms, and many other genomes as well, we have lots of information about the genes within those organisms. And it's often useful to be able to visualize at once lots of different kinds of information about genes in your organism of interest. The way that we do this is through the use of genome browsers. These are web-based tools that help us visualize different features of genes that we're interested in. And next, I'll show you guys a screencast that I made using the human genome browser. Okay, so right now we're in a web browser and we're looking at the website that holds the UCSC genome browser. And this is a fantastic way, a very powerful tool which we can use to visualize information about, in this case, human genes. All the model organisms have their own genome browsers as well. And this genome browser can tell us lots of different things. In fact, if we scroll down each of these drop-down menus, if we select them and tell the browser to display the information in that track, it will tell us something different about the region of the human genome that you're looking at. So we can see that there are lots and lots of things that we can visualize about the human genome. Right now, all we're looking at is what genes are on a given region of the chromosome. So right now we're looking at part of this particular gene, the RB1-CC1 gene. And the boxes show the protein coding regions of that gene. Okay, these are protein coding regions as well. The red line shows us where we are on the chromosome that has that gene, and we can see here that we're looking at chromosome 8, right? So the RB1-CC1 gene happens to be on the long arm of chromosome 8. Now, the human genome browser, like I said, can tell us lots and lots of different information. One of the things it can tell us is where mutations are in any given gene that have actually been found in real humans. And so that's what this common SNPs stands for, single nucleotide polymorphism. Essentially, what the common mutations are around and in this gene that have actually been found in humans, and each line here represents, each vertical line represents a different mutation that's found in or near this gene. And the different color lines just represent different types of mutations. And this is a dynamic browser, so we can actually kind of zoom around, right? We can look at different parts of the gene. We can zoom out if we like. So let's zoom out a little bit. Now we can see this entire gene and where some of the more interesting mutations are found. And there are other things that we can visualize as well, other information that we can put up on the browser window. For example, maybe I'm interested in determining which parts of this gene the DNA is most loosely packed. Oftentimes, loosely packed DNA is easily transcribed, and that's where transcription is initiated. So I'm going to go down to this track, regulation, the encode regulation track, and I'm going to click show that and hit refresh. And this browser tells me in this window here the relative levels of a particular modification that is an indicator of how loosely DNA is packaged in and around this gene. And we can see here that it's pretty loosely packaged around the start of this gene. And that's typical of protein coding genes. We also see a really loosely packed region down here at the end of the gene. So that's the genome browser. We can also zoom way in to the base pair level, right? We can zoom in close enough where we can actually see the DNA sequence. So lots of information you could visualize about human genes using the UCSC genome browser. And we'll come back to this browser at several points in the course. Feel free to play around with it on your own just for fun. It can be a bit intimidating at first, but once you get used to it, it's not so bad. When the genomes of humans and model organisms were sequenced, it confirmed that model organisms do in fact have many of the same genes as humans and other hard-to-study species of interest. 90% of all human genes are present in mice, and 15% of human genes are even present in fruit flies. In plants, 80 to 90% of genes in corn are also present in Arabidopsis. Even yeast and E. coli share some genes with humans. The study of genes involved in gene expression and other processes in these tiny organisms has taught us a lot about processes shared by all species, like DNA replication, transcription, and translation. And this is perhaps the most important feature of model organisms, that they share genes with other species of interest. Because of this, determining the function of a gene in a model organism tells us a lot about what that same gene is doing in another species of interest, like the human. Now, I don't want to give you all the impression that all research is done in model organisms. Research is done in humans. If we want to characterize human genes, we do eventually need to study them within the context of the human body, in human cells, and in human tissues and organs. Studying human cells in culture dishes is one way that this is done. However, a cell in a culture dish is not a human, and sometimes a phenomenon observed in a cell culture dish, for example, a drug that provides resistance to the Ebola virus in cell culture, won't necessarily make real humans resistant to that virus. Many drugs shown to be effective in human cell culture and even in mice simply don't work in humans or are unsafe. This is one reason that the process of new drug development is so expensive and can take a decade or more to complete. Well, there you have it. We've gone over why humans are hard to study, and the characteristics of model organisms, and how they make studying genes easier. Remember to check out the reading for more about each of the model organisms we discussed. Okay, so at this point you should be able to explain how working with model organisms allows us to efficiently determine the function of genes. And that concludes the first chapter of our course. I hope you guys have enjoyed it so far. I know I'm having a lot of fun making the videos. See you all in Chapter 2.